Much of the public discussion surrounding SARS-CoV-2 infection has focused on the interactions between the SARS-CoV-2 spike protein and cellular ACE2 receptors. Now, researchers at the University of the Chinese Academy of Sciences have found that this may only be part of the story. In a recent paper published by the journal Nature, scientists describe a new target for the SARS-CoV-2 virus that is independent of ACE2 receptors, our T cells. One of the critical symptoms of SARS-CoV-2 infection is lymphopenia. Lymphopenia is a condition in which patients exhibit reduced levels of white blood cells called lymphocytes. Lymphocytes are the fundamental defense cells of our adaptive immune system. They consist of natural killer cells, T cells, and B cells. When any of these cells are reduced, it can inhibit our body's ability to protect itself from viruses. Recent reports have shown that marked lymphopenia is observed in 83% of SARS-CoV-2 patients, but little is known about how SARS-CoV-2 effectively dismantles one of our primary tools of defense. Shen et al. Explored this question by first determining which lymphocytes SARS-CoV-2 targeted. Researchers collected blood cell samples from 22 patients with severe SARS-CoV-2 and from 15 healthy donors. They then tested each sample for 3 major lymphocyte cell types to determine any differences in lymphocyte counts between the healthy and infected donors. To their surprise, patients with severe SARS-CoV-2 exhibited a significant decline in T cells only. This suggested that SARS-CoV-2-induced lymphopenia is due to a decline in T cells and that T cells are the primary lymphocyte targets of the SARS-CoV-2 virus. How does SARS-CoV-2 attack T cells? Shen et al. Hypothesized that the virus might directly infect T cells, killing them in the process. To test this theory, they analyzed the T lymphocytes of infected patients for the presence of viral antigens. This analysis confirmed that SARS-CoV-2 antigens were present inside the T lymphocytes and that the virus could directly infect T cells. To confirm this result, Shen et al. Examined lung sections from patients who had suffered from fatal SARS-CoV-2 infections. They found that the lung tissue contained high levels of T lymphocytes and that many T lymphocytes tested positive for SARS-CoV-2 proteins, indicating infection of those T cells, Figure 1. These results were further corroborated by in vitro experiments. Shen et al. Infected 3 model T cell types with SARS-CoV-2, Jurkat cells, MT4 cells, and T cells isolated from healthy donors.